Great, thank you very much, Kim, for this overwhelming introduction. And also thanks, Sarah, for organizing this webinar. This is really a great opportunity for me to be able to introduce myself to the Australian health services research community. As Kim just said, I started in May, just about 4 months ago. And other than getting introduced into the wave surfing here in Australia and some of the other outdoor events, I really have started exploring the environment and getting in touch with a couple of people here in Melbourne. And I hope to be able to get in touch with many others here in Australia to be able to collaborate in the topic of cancer health services research. For today, I actually have kind of a broad overview of the work that I've been doing in the last 10 years, mainly in the Netherlands and Europe, with some collaborations with the US groups and Canadian groups. And I've squeezed in a little bit of work that we're starting up here in Melbourne. And I'd really like to extend that kind of work. So I hope you can apologize for not presenting a huge program from the Melbourne Institute at the moment, because I'm just really starting up the program. So in preparing my talk for today, I thought I'd go have a look at the website from the association and try to look into the definition of health services research. I think if you speak to people about what is health services research, there's all kinds of misinterpretation or misunderstanding of what health services research is. So I thought I'd go to the website and I'll pull the definition that's on the website, and I really like this definition because the definition that you've put forward in the association is about improving the health services that patients receive, and that actually is something I'm really wanting to work on. And this improvement has different components. It has a component of quality of care, accessibility to care, the equal distribution of health gains from the service that we provide, but also about efficiency. And this relates to some of the work that I've been doing in the health economics space, and I've also worked with Kim on that within the health economics group here in Melbourne. So efficiency gains, so both allocative efficiency and technical efficiency. So how can we maximize outcomes given the constrained resources? And I'd really like to this definition. That's the reason for putting this forward. And this will really guide my presentation for today. So some of the key points for my talk today is the first one, I think, precision oncology. And we're talking about genomics, proteomics, but also the use of liquid biopsies, all kinds of molecular markers, and more advanced drug therapy, immunotherapy, targeted therapies. They'll create opportunities, but also challenges for health systems to improve cancer services. Second point I'd like to make, that HTA health technology assessment that we've been working on for the last 10 years has made a significant contribution worldwide in understanding of allocation resources, scarce resources, to maximize health benefits for society. But the drawback of that is that I think HTA traditionally informs about reimbursement and market access to new technologies. It is less capable of evaluating health systems and healthcare delivery inefficiencies. And that's really the angle I'd like to take with the health service research program. And I'd like to share a few of the initiatives, global initiatives, about improving the delivery of cancer services by looking into new services, but also discontinuing low-value services using real-world outcomes. And that's really the future research agenda from my program here in Melbourne. So just to start with a few background slides about cancer, for those of you that are not very familiar with the cancer space. I think worldwide spending, the healthcare spending, is about 5% of total healthcare budgets, and this includes indirect and direct costs of cancer care. And of those 5%, about a fourth, 25%, is spent on drugs. And this share has grown, and there's some evidence from the European, from Bengt Johnson in Sweden. So there's an increase from 12% in 2005 up to 25% right now, which will even grow further to about 30% in 2021. So this increase, and that's in bold, I think it's very important to realize this increase in spending on drugs so far has been compensated by efficiency gains in hospital care. So by creating more efficiencies in service provision within hospitals and primary care, we have been able to keep the budget, the total budget, at the 5% level. So the total budget spending in cancer care hasn't gone up since then. But we don't think this is sustainable. So if you see that further growth in drug spending and all the other components of care delivery that are equally important, we don't think this is sustainable. This huge increase in spending is sustainable. And that's one of the reasons why ESMO, the European Society for Medical Oncology, has initiated a cancer medicines committee, a working group. And there's both a committee about inexpensive drugs, access to inexpensive drugs, the WHO list. But there's also a group that really talks about the expensive cancer medicines and what we can do also as a professional society to keep track of the costs and in order to guarantee access for all. And there's quite a few issues that we've been discussing in the working group, and this is ongoing work, so I won't be talking about it today to a larger extent. But we've seen in Europe substantial variation in price between European countries, but also between hospitals as part of negotiation agreements. And this is something we can work on and think about price differentiation within European countries or between European countries based on their GDP, their affordability. We've been talking about indication-based pricing based on value. So there might be different prices based on the indication, the tumor stream that we're treating. But also initiatives like EU-wide price negotiation with companies. Because now negotiation sometimes is even within the hospital, and by uniting a negotiation power, you might really come up with a good price arrangement. So there's some issues that have been part of the agenda with the ESMO working group. The issue, the 5% budget that we spend on cancer care and the opportunity that we've seen to balance the increase in drug spending has to do with opportunity costs. So how much can we afford, and what is the best way to allocate our resources to get most value for society? So for the health economists joining today, they're pretty much aware of the opportunity cost, but I think I just put in this slide as an illustration. So suppose that we have one course of treatment with pertuzumab for breast cancer that will charge you about $80,000 or euros in this case. Sorry, I'm still in the euro currency. 80,000 euros for one course of treatment. For the same amount of money, you'd be able to do 7 breast reconstructions. So that's an opportunity forgone by investing one course of drug treatment with pertuzumab. But likewise, we could also screen 68 women. So it's lifetime screening 68 women with the same amount of money. So then the question is, where do we get most value for our money? And that's the issue of the opportunity cost. And this is really an issue for most of the health economists, but also in terms of service delivery, where can we add most value across the care cycle? So traditionally, in health economics, we do this kind of analysis by means of decision modeling. We construct decision models, and this is a very simple decision tree, comparing 3 treatments, A and B. They have different profile in terms of success rates. So there's different gains in life years from the 2 drugs, and there's also different price levels. So in this case, treatment B is obviously much more expensive. It might be a targeted treatment, much more expensive, but it generates more qualities or more life years. And we're able to estimate the quality adjusted life year, and in this case, it will be 180,000 euros per life year. So this is just a very simple starting point to understand the decision tree and to understand some of the work that health economic modeling includes in terms of resource allocation decisions. But the issue I'd like to share with you that we've done quite a few cost-effectiveness models, and they've been very successful. I think we will continue using cost-effectiveness modeling, but there's not a solution for all. So I had a little bit of a provoking title during my talk for ESMO 2 weeks ago. Is it future-proof, and what are the issues in cost-effectiveness analysis? Well, the first thing, cost-effectiveness analysis is relevant for some countries, but it's certainly not relevant for other countries. So in Australia, it's very strong tradition like in the UK with cost-effectiveness analysis. But within Europe, France and Germany are not using cost-effectiveness analysis at all. So there's quite a few differences within Europe. The interesting thing is that even there are significant differences in regulatory environments. Countries make similar decisions about reimbursement of drugs. That's one issue. Second issue that we part of the discussion for the last, I think, 40 years since the introduction of the quality is the quality the right measure to be able to measure health benefits, because it doesn't include other relevant components of health, process utility. So there's been quite some effort on using discrete choice experiments or conjoint analysis or multi-criteria decision analysis to capture other components of value. And the second thing is it cannot be used across healthcare sectors, which is in current debate within European institutions. The third one, and that's particularly relevant in terms of access and affordability, is that there's a mismatch between cost effectiveness and affordability. So budget impact is a very important element to include for most of the decision-making settings in European countries. And then 4 and 5, that's the topics I'd like to talk about a bit more today, is cost-effectiveness analysis usually is trial-based on head-to-head comparisons with a therapeutic alternative, so it's comparator. And in cost-effectiveness analysis, and I think that's challenging also from a methods point of view, is not able to deal with multiple lines of treatments, which is very relevant in the cancer space, or with interventions across the care cycle. You usually don't compare surgical interventions to drug treatment. And that might be very relevant in terms of figuring out where we add value across the care cycle. So that's one thing I'd like to talk about a little bit further. And the second one is that cost-effectiveness analysis typically does not consider the wider health system's constraints and consequences. And one of the big things is the clinical trial outcomes from randomized controlled trials typically overestimate real-world outcomes. And the second thing, and that's very important also for the ESMO, that decisions are made without considering the real-world effects, such as disparities in access. And I'd like to share a few papers with you on disparities in the delivery of cancer services. So the first one, this is, I'm sorry about the European perspective, but this is a map of Europe in terms of the differences in incidence, cancer incidence, and cancer mortality. So you can see from the map here that the cancer incidence is pretty high in the North European countries, West European countries, and much lower in the East European countries. But mortality on the other side, it's much higher in the East European countries. So there's disparities in terms of the cancer incidence and the quality of the services that we provide in order to prolong survival. So that's one disparity. A second disparity in the European context is the time lag between a market approval decision from the European Medicines Agency and an actual reimbursement decision within the member state, because that's different decision bodies. European Medicines Agency is across Europe, and they can decide about approval of a drug. And that means that it's usually immediately available in the UK and in Germany. But for many other countries, there's a time lag ranging from 116 days in Denmark to more than a year and a half in the Czech Republic. So there's disparities in terms of access to services, to new drugs for some of the European citizens. And this might be, I'm pretty sure, because I've seen some of the data from at least Victoria and Australia. This is pretty much similar for Australia. And I've seen similar cases in Canada, for instance, with a different legislation in different Canadian provinces. And the third disparity I'd like to share with you is access to trials. So we know that most patients only can get innovative new treatments by participating in a clinical trial. So the number of trials that run in a particular country guarantee access to new innovative drugs. So this is a map that I've made from clinicaltrial.gov, and you can present the number of trials, and this is prostate cancer, advanced prostate cancer trials. The number of trials that enroll patients in each of the countries, with the UK on top at 47 different clinical trials. So the likelihood to be enrolled in a trial in the UK is much higher than in the East European countries. So innovative drugs, new drugs that come into the market, will easily be available to people living in the UK or in the North and the Western European countries, but not so much in the East European countries. So that's another element of disparity in access to drugs. And then if we make a decision to reimburse, and you're not able to read this slide, but there's a reference here that you can download. These are the European countries in which all the decisions are tabulated. So the blue means this is a reimbursement decision, and blue, this is free of charge. There's no copay from patients. But the red means it's available only at full cost, and this is the 5 different drugs for breast cancer: trastuzumab, pertuzumab, chemotherapy. So the blue, that's usually the North European, Western European countries that have it freely available. There's no copay for those drugs. And this is usually the East and the South European countries, in which is only available at a copay, a significant copay up to 100%. So that's a reimbursement decision. But there's also a question about availability. It might be reimbursed, it might be a formal decision of reimbursement. It doesn't mean that it's also available. So this table also presents the availability of the particular drugs in those countries. And you see a similar trend that in the North and the West European countries, it's also available. Most drugs are available, but in the South and West European, East European countries, it's not even available. That's the black dots here. It's not even available. Even though there might have been a reimbursement decision, it's simply not just available. So this is also something that we have to keep in mind if it's about reimbursement decisions and disparities in cancer services. Then I'd like to share 4 examples that there's quite a few bit of evidence on real-world variation in outcomes and services across the continuum. So the first 2 papers is the evidence of variation in outcomes. So this is a recent paper in the British Medical Journal reporting that after market entry, cancer drugs rarely show the benefits in overall survival or quality of life in randomized trials. So there's this mismatch between trial evidence, which is used for reimbursement decisions, and the actual real-world evidence. A second paper, this is the ESMO magnitude clinical benefit scale, and they evaluate drugs that come into the market based on clinical trial evidence. And they conclude that less than a third of the contemporary RCTs with a statistically significant difference meet the ESMO thresholds, which is only 15% of all the published trials. So that's an interesting thing to realize. And then in terms of service provision, and there's quite a bit of evidence on variation in the system between services provided, and this is a paper from my own group in the Netherlands. It's comparing different hospitals in immediate reconstruction, breast reconstruction, in patients with DCIS and with invasive breast cancer. You see there's quite a bit of a range from 0 to 80% of patients that receive an immediate breast reconstruction following surgery. So that's quite a variation in services that we provide to patients. But also in terms of new guidelines, new adherence to new guidelines, we found significant variation in the use of active surveillance. It just takes time to be able to implement guidelines, to be able to adhere to guidelines. And policies in the case of breast cancer to achieve consistent and higher rates of active surveillance should receive priority. So I think these slides actually present some of the evidence about variation in the system that are usually not captured within an HTA framework or within a deliberative process of making a decision about reimbursement. And I think pretty much important for us to realize also in developing a health service research agenda, because this is really where health service research comes into play. So in the next part of my talk, I'd like to focus a little bit into the issue of genomics, genomic sequencing, the use of molecular diagnostics and liquid biopsies, because this is really taking off, and there's quite a few research efforts going on worldwide in the use of new innovative technologies like NGS and whole genome sequencing to improve services to patients, to improve targeting of drug treatment. And this is a challenging topic as well, because there's just been a special issue within Value in Health, which was organized by Catherine Phillips from San Francisco. And she concludes what we all know, there are rapidly developing technologies, but the assessment of the societal value remains very challenging. There's too many problems and too few solutions. So there's quite a bit of methodological work to be required here. In my own research program, we've done quite a bit on the use of liquid biopsies, so the use of different tumor markers in blood or tissue fluids like urine to either screen or to monitor treatment progression. And we've seen, if you read Annals of Oncology or some of the oncology papers, there's quite a few papers coming out about the use of ctDNA, circulating tumor DNA, tumor-derived extravascular microRNA, all kinds of new tumor particles that might be useful for monitoring treatment progression, to inform treatment switching, for screening of cancers. And all those technologies, because it's a very relatively cheap, you can just draw blood and do tissue analysis or blood analysis. So it's a relatively cheap way of collecting tumor material. You can frequently administer those tests and really use it for continuous monitoring of disease. So there might be some health economic impact of using those liquid biopsies. I'll show that in a few minutes. The most experienced at the moment, so this is all research which is ongoing, but the first experience were with the use of circulating tumor cells. CTCs are cells that circulate through in the blood that are detached from a primary tumor and are a sign of distant metastasis. So you could be able to analyze the CTCs from blood to predict progression-free, there should be free survival and overall survival. And there's quite some convincing evidence on that. This is a Kaplan-Meier plot on the prostate cancer. You can see that if you're above 5 cells per 7.5 mil, then you have a least favorable progression of your prostate cancer. You can separate, you can stratify patients with a favorable and an unfavorable prognosis. And this is evidence that's been out there for quite some time. So this is some of the new technologies that we're working on. And there's a great need to think about and to analyze the potential health economic effects and the potential impact on health services. And this is a recent consensus statement from a group of experts about the need for further evidence development in the metastatic prostate cancer space. So let's go to the metastatic prostate cancer space and just have this first slide which would present you some of the lines of treatment that we can administer here. So this is all metastatic castration-resistant prostate cancers. So we've all had first-line treatment with hormone therapy, and it starts to progress. And usually first-line treatment after in progressive disease might be docetaxel. It's a relatively cheap drug. But then if it is not responding, you might switch to a second-line drug, which could be abiraterone, which is anti-hormonal therapy, or enzalutamide, also an anti-hormonal therapy. And these are much more expensive, about 30,000 euros per cycle of treatment. And finally, there's also cabazitaxel, 64,000 euros per cycle. And there's some other alternatives, radium, for instance, or immunotherapy in prostate cancer. So the interesting thing that in the original approval, I mean that the reimbursement decision, for instance, for cabazitaxel, were as a third-line treatment or second-line treatment, only after progression, only after first-line treatment didn't really work. But now trials are ongoing to really use cabazitaxel even in first line. So you see this all, the use of these different drugs and the different lines of treatment is very dynamic. And there's some very strict, there's some guidelines, but I think it's very important to really look at the real world evidence on how those drugs are being used and what trial evidence is there to really change the patterns of use and the sequence of use of those drugs. But let's talk about the testing. So we do have a couple of biomarkers that could predict or stratify patients that are likely to respond from a particular drug or less likely to respond. So this is an ARV7 test receptor on a circulating tumor cell that can be analyzed. which is an indication of responsiveness to anti-hormonal therapy. So if you're resistant to ARV7, you might not respond to anti-hormonal therapy. So you can do the test, and if there's a high likelihood that you're resistant to anti-hormone therapy, you wouldn't go to either abiraterone or enzalutamide, because it will probably not be effective in this group of patients. You might go to either cabazitaxel, or you even go to cabazitaxel. So that's the idea of ARV7 testing, just a single biomarker at a given point in time. And there's some evidence. This is not a health economic model, but this is just a simple budget impact model from the US, from Baltimore, in which they modeled the potential savings, downstream savings, of implementing an ARV7 test, just to stratify patients that should receive anti-hormone therapy and patients that shouldn't receive anti-hormone therapy. So the prevalence of resistance in this group, and this prevalence depends on previous cycles of treatments. So the biology changes. So if you didn't have any anti-hormonal therapy in a previous cycle of treatment, so only docetaxel, you're in naive patients, there's only 10 to 15% resistant to anti-hormonal therapy. So there's a high likelihood that it might be very effective. But if you had one course of anti-hormonal therapy, this prevalence, the resistant number of patients that are being resistant to anti-hormonal therapy increases to 30%. And after post-abiraterone and enzalutamide, you have 2 courses of treatment, it goes even up to 50%. So this prevalence really changes depending on the drug that has been used in the previous cycles. So you could do a simple calculation with the cost of the test, the ARV7 assay, cost that you can avoid, and the total savings for the US health system will be $150 million. Just by implementing a new test. So you account for the new testing, this is the saving. So there might be some potential effects of implementing an ARV7 test. But then it becomes a bit more complex. So this is the real world evidence, and this is work that we're starting up here in Melbourne with the WEHI and Royal Melbourne, and some of the institutions around Australia, because everyone contributes to this data registry, the EPAT registry, which is led by Ben Tran and Peter Gibbs. So there's about 700 patients in the registry at the moment, and we've been able to identify treatment patterns. This is real-world evidence. I said first-line treatment in most patients is docetaxel, and then the second line will be an anti-hormone therapy, it's 80, and there might be even a third line cabazitaxel. And there's even more lines of treatment in a specific part of the patients. So there's a group of patients that receive docetaxel in the first line, abiraterone, and also quite a few patients that receive enzalutamide in the first line. And here, I mean, if you think about testing, the use of molecular testing to guide treatment decisions and switching, it becomes even more complex. So this is a typical pattern that we can identify. So start with anti-hormonal therapy, 2 courses, and then cabazitaxel. So the decision to prescribe either enzalutamide or abiraterone depends, of course, as shown before, on the resistance to the treatment. So if we go back to the previous slide that I showed you with this simple, the 30% of the patient are resistant to anti-hormone therapy, we can extend it. So we can use the same test at every decision point here, and we can really model, we can map the care pathway with all the sequences and the different tests that we're going to prescribe. So in this case, it starts at 15%, so they are naive patients, 30%, 1 course of treatment, and then up to 45% for 2 courses of treatment, resistant to anti-hormone therapy. And this requires a bit of a different approach, and that's what I wanted to share with you today. I'm not going to talk about the specifics of the diagnostics and how you could use it. I'm just thinking about health services and what we can do to inform that kind of decisions, and how we should be able to analyze the value for society of adding these testing strategies. So that's why we are using different methods, because this is not a simple head-to-head comparison, that's not trial-based, because we are using the real-world evidence from a registry like EPAT. We've been using other registries, so we know what are the treatment decisions that are being made. We potentially can also link it to biomarker profiles and try to come up with a a simulation model in which we simulate the care pathway. We simulate every single patient, all patient level. We start them here. We simulate a first-line treatment. We can simulate the tests that are being administered. We can simulate a decision to switch to second-line or third-line treatment. So this modeling approach is all patient level. We need data from every individual patient about individual characteristics, the treatments prescribed, admin data, claims data, cost of the procedures that we prescribe. And then we can build a simulation model to be able to analyze different patterns of care across the care pathway. And this is just a very simple model, a discrete event simulation model that we built within a trial, which is called a CTC stop. It's a trial which is led by Melbourne in the UK. This is the use of circulating tumor cells to inform a switch to a second-line treatment, which is in this case, which is Capsicum in patients that receive docetaxel in the first line. So this is, I just wanted to illustrate you the idea of simulation based on real-world data and evidence from clinical trials to be able to inform about the different care pathways and inform about the potential health benefits and economic benefits across this pathway, the sequences of treatments. And this is a bit different than the work that we've been doing so far with head-to-head comparison of just two drugs within a trial-based environment. So again, this is using real-world data. So this is just how it works. So this is a simulation interface. So we built an interface, and we've done it with a group of physicians in which we can simulate different testing strategies using CTCs or just ordinary PSA testing or bone scans. And we can manipulate that. We can identify the sensitivity, specificity, so the diagnostic performance of either tests. So it could be CTCs, but potentially if you think about future developments in the genomic space, we can also think about ctDNA. So if there's some preliminary evidence of ctDNA and the diagnostic performance, we can simply identify the diagnostic performance and change it in the model here. We can do the same for the PSA, which in this case is the comparator diagnostic strategy. We can add, based on clinical trial evidence, the quality of life estimates from trials using docetaxel in the first line, Capsicum in the first and the second line. So that's all kind of population of our discrete event simulation model, including the costs that we obtain from the registries and the claims data. So that's the input of the model. And then there's a very important thing to realize. We can also include the physician preferences. So we might inform a physician to switch to second-line treatment, but there might be very good reasons why a physician is not willing to, or a physician with a patient together are not making that decision to switch because of toxicity. There might be very good reasons. So we can simulate a case where every decision is followed according to what we recommend in terms of the diagnostic strategy. We can also include other variables, other preferences in order to make that decision to switch to a second-line or third-line treatment. So this is the interface, and I'll show you a bit of the simulation because this is the simulation model where you could include, that's the good thing of the simulation, you can increase the number of patients that you're simulating. Very easy, it's just one button, and very cheap also. You can change the performance, you can change the costs of the drug treatment, and you can start simulation. And this is what you can really say, and this is very intuitive also if you talk to physicians about what's going on here. So this is very intuitive in terms of the sequence that you, I'm not sure if I can show you, I can show it again. So you can see, it's switching back, I'm sorry. Anyway, you could see that we were simulating about 30,000 patients, and you can see all 30,000 flowing through the model based on prior distributions that we obtained from the registries and the clinical trial evidence. So I think as a conclusion, what are the implications of those developments for services and for evaluating societal value of new innovative technologies like diagnostics, but also new pharmaceutical agents that come into the market. So I think the implications, from my perspective, that I'm very, very open for any other feedback and discussion about it, is that we use real-world data instead of clinical trial evidence alone, because that allows us to include practice variation and constraints. That's one thing. The second thing is that we need to develop new methods to personalize healthcare delivery. So we can think about simulation optimization using patient-level data. And we can also use the same modeling approach to assess new innovation, early stage, by looking at the whole cycle of care. So where does it add value? That's I think the implications of the developments that I just shared with you. So just as an example of that work is a project that we've run on follow-up care, survivorship care. And this really is more about the service provision and what's the optimal follow-up program that we can develop for patients. So this is, we all know there's different nomograms, and this is a nomogram based on the Netherlands Cancer Registry data. It's about 40,000 patients that we have analyzed based on the different characteristics, and it could provide you the time-dependent probability of recurrence between 0.5 year and 1.2 or 3% in third year. So based on the time-dependent recurrence rate, you might think, well, we need the most intense follow-up here. But this is very much dependent on the risk. So it's risk-based, so you can identify different strata that require more intense follow-up. So that's the work that we are now doing, because the current guideline that used to be 4 times in the first year after breast cancer surgery, and 2 years in the second year, and then 1 time a year thereafter, has been revised to one visit a year, 1 MRI mammogram every year. That's the current guideline for the next 5 years. But that follow-up scheme might not be required for every single patient. So we can reuse this kind of prediction model to optimize the survivorship program for patients, so the follow-up program. But it's not easy to do that with standard methodology. So we can do that with decision trees. We can make that decision at every point. Do we need to have an MRI, or do we need a mammogram at this point? And we go to the next one. But because we have different decisions to be made, it will be very complicated to do it in this modeling framework. So we think about modeling sequential decisions, because those decision trees become very complex. It's very hard to handle time within those sequential decision trees. So we were exploring the use of market decision processes in this case. And I'll just show you the result. I won't talk about market decision processes, but what we were able to identify in this case, we had 4 different age groups based on the risks of local regional recurrence following breast cancer. And we were able to, because those risk groups or age groups have different risk profiles. So the risk of a recurrence, a local regional recurrence, or a second primary is much higher in the lower age group than in the higher age group. And using this kind of partially observable market decision processes, we can really optimize the allocation of resources. So this M means that's MRI, or follow-up visit with an MRI or mammogram. So in the lower age group, the recommendation would be to we don't need any follow-up in the first year, but then we have a very intense follow-up scheme. Every half year, we need a patient to come back in hospital undergoing mammogram for routine inspection of recurrences on second primaries. But in the other groups, it is a very intense follow-up scheme. But in the other groups here, more than 70 years, there's only 3 after 1.5 years, 2.5 years, and 3.5 years. So this is how you could use, just an example, how you can use registry data to inform about recurrence risks and risks of second primaries, and optimize, from a hospital perspective, optimize the scheduling of follow-up visits and the use of mammography or MRI in detecting local regional recurrences in second primaries. So there's also it's more technical efficiency in terms of how can we maximize outcomes given a constrained budget within the constrained resources within the hospital environment. So this is the type of work, also in terms of future outlook. This is the type of work that we're trying to develop. This is an initiative that we're working on between ESPOR and the European Cancer Organization, based on the essential requirements from ECHO. So ECHO essential requirements define the essential cancer services that need to be provided across the care cycle. And with the initiative between ISPOR and ECHO, we try to do a health economic perspective across the care cycle, following those essential requirements. And this is just an initiative which is ongoing, and we hope to be able to identify where we can really add value in terms of service provision. So the final few minutes, I'd like to talk about a different perspective. So far, and most of the work that we've been doing is taking a causal perspective. So we have a set of determinants and an outcome. But some of the work that we do is much more complicated. And healthcare really is a complex system. So we probably need to take a systems perspective at some point, that we need to consider different inputs to the system, different outcomes, and a system as a whole with an independent behavior of the system. And so this systems perspective, this is really taking off. There's a couple of initiatives thinking about more systems, implementing more systems thinking to health technology assessment. And the example I'd like to show you, and this might be very interesting also for the Australian context with the Australian genomics, some of the other initiatives in Australia on genomics. This is about implementing genomic sequencing in oncology in the Netherlands, where the discussion is, do we need to have one facility for sequencing all the tumor material, tumor biopsies, and then feed back the information to all of the hospitals, and every patient is being treated in a hospital, being referred to a primary hospital, biopsies being taken, sent for sequencing, and then we expect the results to come back to the hospital for treatment decision making. So that's the idea. So the normal procedure would be use a panel, then target therapy, and look at health outcomes. But here we're going to implement whole genome sequencing. We take the biopsy up to the central sequencing facility, which is stored, and then we can feed back that information to the clinical decision within the hospital. So this is in the local setting. Well, you can imagine that there are quite a few issues to consider here, because there's quite a bit heterogeneity between patients. There's quite a bit heterogeneity between hospitals. So there are agents between physicians, expert levels between hospitals, volume of care, and this is all nonlinear. So it's not easy to model that in terms of a cost-effectiveness model. So here in this case, we're trying to implement a systems-wide model where we really look into referral patterns. And this is one of the first examples of data, real-world data on referral patterns on stage 4 non-small cell lung cancer patients. So this is all patients, all referrals of patients between hospitals in the Netherlands. So this is all 79 different hospitals in the Netherlands. And you see there's 30% of all patients are being referred to another hospital for one service. It could be either immunotherapy, or it could be palliative care, or radiation oncology. So this is 30% of patients that are being referred at some point. So it's quite a few referrals between hospitals in terms of delivering services. And this also might create delays in treatment. So this is another figure that we got from the same data registry from the Netherlands Cancer Registry, where we mapped the delays between diagnosis and the start of treatment. And I just, for clarification, I just illustrated here. These are the general hospitals. We have a series of academic hospitals, teaching hospitals, affiliated to medical schools, and these are the general hospitals. And you can see there's a substantial variation, up to 40 days between the start of the treatment after diagnosis. So that variation needs to be taken into account. Also, if you're about implementing whole genome services, so what does it mean for some of the patients in a country in terms of getting access to those services timely, so we can really change their treatment pattern in a timely way. So this modeling, what we do. This is another quick simulation. Takes a very different approach because this is really a systems modeling approach where we implement this as an agent-based simulation where we identify the different hospitals. There are agents here. The reds are the hospitals in a country. The blue is a central sequencing facility. And all the dots are our patients that are being referred from the one hospital to another hospital. And if they're referred, we of course we have to map the care pathway, and that's what you can see here. So this is a care pathway in which there are different treatment options, and this is very similar to the case I showed you before with the prostate cancer. I'll show you to the next slide because that makes it easier. So this is the care pathway that we map within each hospital, and we can do that for every single hospital. So there's guidelines based on the treatment after diagnosis. There's different treatment options: immunotherapy, targeted chemotherapy, and we can map this care pathway within the hospital, and we can fill that pathway with specific data from the hospital. And this is all underneath this systems model. So we have both on a very high level, societal level, the hospital in a model, but we can also map and model the service provision within the hospital. And this is the way of thinking in terms of the systems approaches, and that might be relevant for something like whole genome sequencing, but also for proton beam radiation therapy, in which there's only 1 or 2 centers in a country, at least in the Netherlands, or some large-scale infrastructure. So that's the more systems approaches. That was what I wanted to share with you. So I think to conclude, I wanted to cover these 4 topics. I think precision oncology creates the opportunities, but also challenges. I've showed you some of the challenges in terms of adaptive treatment pathways and how can we add value to that. Also the systems modeling. I think we all agree that HDR has made a significant contribution, but we need to do more than that to inform a health policy about inefficiencies in the system. And that's why we need health service research, and we need real-world data registries to be able to analyze the inefficiencies in the system. And I think with that, I think I can conclude, and we have still a couple of minutes for Q&A. Thank you so much.
Great, thank you, Maarten. I think I just need Sarah to start my video again. But yeah, we'd now like to invite questions from any of the attendees. You can use the Q&A tab. If you scroll down and put your cursor on your bottom of your screen, you click the Q&A, and you're welcome to type in a question. So please do that. While we're waiting for that, I wonder, Maarten, if you had any reflections at this point about the quality of Australian data. Obviously, to build these type of models that reflect kind of the current treatment pathways or the current use of different tests and pharmaceuticals and so on, it relies on, you know, a reasonable quality of data. What's your sense so far, having been in Australia, about what our capacity might be to provide the type of data needed for these sort of modelling exercises?
So I think I'm just here for 4 months, so I'm really exploring the quality of the data. But most of the people have told me that Australians are not satisfied yet with the quality of the data. And with that in mind, I went to many people, and I must say there's quite a few data sets that we can use. As I said, there's quite a few data sets within. So the EPAD data registry is a very interesting one to start with. There's a few more. There's also a couple of data registries, I think, very important for quality improvement programs run through Monash University. And I think there's quite a few initiatives. So people are pretty much aware that there needs to be done some extra work, but there's very much initiatives and a lot of activity to really link data sets and to be able to analyze and use that data in I think within a few years. So I think there's quite some opportunity here within Australia. That's the first reaction I could give to.
Okay, great. And also, I guess the other thing I was keen to explore a little bit further is it was really striking that the issues around these multiple lines of treatment, right? And we can see in our Australian system that when we evaluate a new pharmaceutical, we're very much, or a new test, we're evaluating it sort of one at a time. And I think we see in things like breast cancer, even breast cancer, prostate cancer, after a while, we can look back on our decisions and see that it is really a complex web of treatments that have been regulated. And, some are first line, second line, third line, so on. And we look back and wonder sometimes whether we got it right, I guess. And a lot of those decisions are being referenced off each other. And so I guess I was really interested to see your particular your thoughts around really evaluating a system and that line of the whole complexity of the line of treatment. My question really is, at what point do you think it becomes useful to initiate that simulation modelling? Because I guess we start with just one treatment and one test, and then we add another and then another and another. And I think we can look back and go, that would be really useful to do now. But at what point do you think it's helpful to start initiating these sorts of modelling exercises on the real world data?
So that's a very good question. I'd like to open up for a broader discussion, really. So I just identified the issue, and I just proposed some of the methods that we can work around with. I don't think we can replace the current HTA processes, so we still need to do this head-to-head trials and have a proper judgment of the evidence and the economics for single agents that come in the market. But I do think there is a place for disease models for particular diseases. You might do that within the tumor stream or within a therapeutic area. So disease models that really use real-world registries and are so flexible that we can also extrapolate the effects of new innovation. And this requires a lot of collaboration. It requires data sharing. It requires sharing of the modeling that we do. Yeah. It's not an effort that we can do within a single group. It's an Australian effort, at least. And maybe it should go beyond that. And this is a different way of thinking in terms of HTA and informing health policies about new innovations in healthcare.
Yeah. I mean, it does, I think the point at which we have thought about this in the past is sometimes when we get a particularly complex disease pathway that's become very expensive and is really densely populated, and we get to a certain point that we think now might be a good time to go back and review these treatments and sort of think about a single model that we apply all the treatments we've currently got to. But I mean, the use of the registry data was really interesting. So yeah, I think there's definitely a lot of scope for this in Australia. I've got a question come through here from Tanya Pigeon. So really, she's just encouraging reaching out to she's given, and we'll give you a copy of this Q&A, Maarten, but she's made some suggestions, I guess, about some other research groups and clinicians that they would really like to have you make contact with. So I think that's a good idea. And I guess it does strike me that we're small in Australia, right? So do you have any advice? The Netherlands is not a huge country either, about the benefits, I guess, of gathering up our power or gathering up, how do we as a small country position ourselves in these global markets to kind of make better decisions when we're small?
Well, the population is quite the same. It's about 17 to 23 million.
Yeah.
It's bigger. But the country is, of course, much bigger. Maybe to follow up on Tanya, I really like to catch up with you and the team in Wollongong. But also an invitation to any other participants today. I'm really looking forward to catch up with you and interact and see where we can work together. So just let me know by email or so. Just going back to the Netherlands, and also in terms of the data sharing and the data registry. So you might think that we're a little bit ahead of you, but the data that I shared with about the lung cancer, it took us half a year to get that data from the registries. Even there, they have to go into the hospitals to get the real therapeutic decisions and the therapy areas and link it to the cancer registry. And this is similar to Australia. This is confidential information. The hospitals do not want to share treatment information. They don't want to share outcomes in public. So the only thing you can do is have a good governance structure and try to use outcomes data, and that's the experience that we have in the value-based healthcare agenda in the Netherlands, to share outcome data within a small group of hospitals in a trusted environment to work on quality improvement programs and not on some sort of public reporting, because it doesn't work. It's just about quality improvement. And you need to engage with clinicians to improve the quality. But again, I think you think you're behind. I don't think that's too big of an issue. There's lots of initiatives going on here. And even in the Netherlands, it's a smaller country. It's easier to travel around. But it's taken quite a bit of time to work through the registries and to make this available. What sometimes works,
yeah.
I'm just thinking about what could work and what would be great for maybe this society to work on, also with health policy and governments, if there's a government incentive to implement a registry. So we've had it for immunotherapy and melanoma. It was a mandatory registry. Hospitals that were willing to provide the treatment were mandatory, obliged to participate in the registry. So that could help also to collect the real world data.
And it does strike me that some of the health services research that you're suggesting, things around, understanding optimal follow-up, understanding, which tests and pharmaceuticals are most effective at which points in the treatment pathway. Some of that investment, I guess, in that data is a little bit of a significant upfront investment that will then pay off later. And I guess what's been your experience in the Netherlands about, the willingness of hospitals and governments to make those investments, in either the data or the models that really will help them to make a lot of future decisions better?
There's in the Netherlands, there's a few good experiences led by professional societies. So there's in the hematology space, there's a big initiative. All the hematology oncologists together have formed a registry, and if they unite, that can bring funding either from government or from industry, because there's a shared interest. We now see the same for the prostate cancer registries. This is a professional society that has taken the initiative to develop the registry. So it's very powerful to work with the clinicians to get this done.
It does sound like the clinicians themselves can be, are really critical in driving a lot of this research through, if they can buy in and have an interest and really see the value of what the research can produce. That's really, really helpful. Yeah. Any other questions from the group at this point? We're assuming they're all just brilliantly satisfied with your presentation, Maarten, and blown away. So great. I think if there's no other questions at this point, please feel free to follow up with Maarten directly. He's new in Melbourne and Australia, so I'm sure he'd love to receive any emails or introductions from any of the group here. Thank you. I guess, do you want me to hand back to you at this point, Sarah?
Only just to remind people, I will send out the recording and
okay,
information, so I can attach Maarten's email address as well if people want to get in touch with any questions. Yeah.
Great, excellent. But thanks so much, Maarten. Like the Health Services Research Association Australia and New Zealand just were so pleased to have you deliver a webinar for us. We're looking forward to kind of increased involvement with the organisation, and thanks for a wonderful talk today. It was very thought-provoking.
Thank you very much. Much appreciated.
Thanks, Maarten, and thanks, Kim.
Thank you.
Everybody.
Thanks.